This is the Professor, CBMD PhD, and today is Tuesday, August 18th, 2026. It is 8:28 and 13 seconds, p.m. Eastern Standard Time, and today's research, Aquatic, Toxicology. So let's hop to it here. Low-dose constant drip, pharmaceutical, personal care pollution, impacts aquatic life. Criteria for assessing toxicity of common chemicals. Ignore ecological side effects. Date November 14, 2017. Source: Carey Institute of Ecosystem Studies. Traditional toxicity testing underestimates the risk that pharmaceutical and personal care product pollution poses to freshwater ecosystems. Criteria that account for ecological disruption, not just organism death, are needed to protect surface waters, which are under pressure from a growing population and escalating synthetic chemical use. Wastewater treatment plants are not designed to remove the chemicals found in pharmaceuticals and personal care products. Instead, these chemicals enter waterways where their effects on aquatic ecosystems are largely unknown. Some 15 years ago, a landmark study found pharmaceuticals and personal care products in 80% of streams sampled across the U.S. Additionally, research confirmed similar patterns globally. The majority of these compounds are understudied, unregulated, and/or deemed low risk. Yet there is a growing body of knowledge that pharmaceuticals and personal care products disrupt aquatic ecosystems and even at low concentrations. For example, when exposed to antidepressants, fish exhibit altered feeding behavior and can become more aggressive. SSRIs have been found to make tadpoles more susceptible to predation. Antidepressants and amphetamines can alter the timing of aquatic insect emergence. The bottom line is that even at low doses, pharmaceuticals and personal care products have the potential to disrupt the ecology of a system, leading to broader environmental consequences. Many of these compounds are pseudo-persistent because we are constantly adding them to our rivers and streams, but there are few studies on how they impact aquatic ecosystems. To assess toxicity, chemical compounds are evaluated with an LC50 test. Organisms of a single species are exposed to increasingly high concentrations of a substance until 50% of the experimental population dies. This concentration is used to set environmentally acceptable limits. Single organism lethality does not account for the diversity of species in nature. Bioaccumulation are non-lethal but disruptive impacts that compromise ecosystems. Traditional toxicology tests do not account for the effects of compounds mixing. The reality is that pharmaceuticals and personal care products rarely appear singly in the environment. Consider the lengthy ingredients list in products like toothpaste, deodorant, shampoo, ibuprofen, or antihistamines. This synthetic chemical cocktail flows into waterways where the compounds are free to interact in ways that were never tested in a laboratory. If you went to your doctor and mentioned that you were taking 30 different drugs, your doctor would likely tell you to stop, and yet this is what is happening in the environment. The bugs, fish, plants, and algae, they are all exposed to this mixture of drugs, and we don't know the impacts. We should think about how pharmaceuticals disrupt ecosystems, not just whether they kill things. The concept of ecologically disrupting compounds mirrors that of endocrine disruptors. Take, for example, bisphenol A, BPA, a chemical found in plastic products, which can act as a hormone in the human body. If someone consumed BPA, it would take a large amount of the compound for it to be lethal. However, even in low concentrations, it can interfere with the function of the endocrine system and cause adverse health effects. Like endocrine disruptors, which impact the human body in low, non-lethal concentrations, we argue that low but persistent concentrations of ecological disruptors cause significant, yet not immediately lethal, effects in the environment. One way to do this is by adding low concentrations of pharmaceuticals to artificial stream communities, which include algae, bacteria, and insects. We monitor the system for any changes. We are not looking to see if all the bugs die, but do they emerge sooner? Does the bacterial community look significantly different? Does the rate of photosynthesis change? Those things are signs of ecological disruption. While this test is key to better understanding the ecological effects of pharmaceuticals and personal care products, with so many different chemicals entering the environment, it would be impossible to test and regulate them all. Instead, we need to focus on the source. Chemicals that we use in our everyday lives enter the waste stream, which is not treated sufficiently. There is a dire need to support our wastewater infrastructure and treatment facilities. Reducing use of products containing these compounds is also important. We want scientists and funding agencies to undertake research focusing on the effects of pharmaceuticals and personal care products in aquatic environments and the extent to which these substances disrupt ecological processes. Ultimately, we hope that a greater scientific understanding of this issue will lead to increased public awareness of the need to keep these compounds out of our ecosystems. And this video was created for educational purposes only. Hope you enjoyed listening. My code name is Will. I'm Professor Christopher Brunet, leader of research scientist, ufologist, psychic computer technician, computer programmer, philanthropist writer, mathematician artist, photographer film producer, chef animal and nature lover. My passion is world peace. Have a great day.